Alright, in this unit we're going to be looking at protein synthesis, which just simply means how do the cells make proteins? Okay, how do organisms make proteins? There are two parts to protein synthesis, transcription and translation. This first part will look at transcription. The second part will get into translation. So here we go. Let's start looking at this. Why do we need proteins? We've looked at this in a lot of different topics that we've covered. First of all, remember proteins are macromolecules. They're building blocks. Their monomers are amino acids, and there's a lot of different reasons why we need proteins. Anything from enzymes to help for cell processes and functionality, to hormone production, to structure, to building of muscles, cartilage. There's millions of different reasons why we need proteins. We know proteins can form filaments. The sequences of nucleotides in the DNA contain the codes or the information to build those specific proteins that are needed by that organism. And there are two major types of nucleic acids. Remember, proteins are polymers of nucleic acids. There are two major types of nucleic acids that are needed. Hopefully you remember, DNA is one and RNA is the other. We've looked at how these are formed. But we're going to get into just briefly comparison. Remember, DNA contains the sugar deoxyribose. It is double-stranded, so it is a very large, complex polymer of a nucleic acid. It uses those bases, adenine, thymine, guanine, and cytosine, to build the structure. They are held together using hydrogen bonds, and it is so large and complex that it cannot pass through those nuclear pores that are found in the nuclear membrane, so it cannot leave the nucleus. Whereas RNA is made of ribose. It's going to be smaller. It's going to be more simplified. It is single-stranded. It uses the same bases except thymine. It does not use thymine. It uses uracil. It adds to the simplicity of it. And since it is going to be a small packaged instruction manual, if you will, that's written by the DNA, then it can leave the nucleus. Okay. Remember, we've also talked about the three major types of RNA. There's messenger RNA, there's transfer RNA, and there's ribosomal RNA. In messenger RNA, we've talked about this before. Remember, that's the RNA that is coded by the DNA in the nucleus. It carries that message, or it carries the instructions from the nucleus into the cell. It'll locate ribosomes, usually on the rough ER, and then it will carry that message to those ribosomes to tell the ribosomes what protein needs to be made. The transfer RNA are going to carry those amino acids, and remember, amino acids are the building blocks of a protein. They're going to be the ones that go fetch and carry those amino acids to the ribosome that are going to be needed to build that particular protein. And then ribosomal RNA, yes, remember we've talked about this before, it is found in the ribosomes. It's a part of that ribosome, and it actually moves like a conveyor belt to help attach those amino acids in the right sequence to build the proper DNA, or to build the proper protein. Here is a overall picture, if you will, of how protein synthesis works. You've got transcription that's going to occur. That's going to be the process where DNA makes RNA and it's going to transcribe that information. And then once the RNA has that information, it is going to take that information to the ribosome, where it's going to be translated into protein. Okay, this is the overall picture. Okay, can't stress this enough. Remember, those amino acids, those are the building pieces that of the nucleic acid that helps build that large polymer protein, that large polypeptide. And we're going to be looking at these amino acids, where they come from, in a minute. But remember, DNA it has the coded information to make the proteins for the organism. It is very large and complex, so it cannot leave the nucleus, and that's where this messenger RNA comes into play. So how transcription works. The coded information that's going to tell the cell how to make particular proteins are going to be transcribed. Okay, it's going to be transcribed from the DNA into a piece of messenger RNA, into a small little instruction book. This little piece of messenger RNA is going to be able to leave the nucleus and carry that transcribed message, that instruction manual, if you will, to the ribosomes. So the ribosomes know what protein to make and how to make it. Here's how it works. We've kind of looked at this process before. Remember, DNA is going to be a part of the DNA strand, not all of it, just a part of it is going to be unzipped by helicase, the enzyme helicase. When it does this, okay, now you have the exposed bases that aren't attached to each other because those hydrogen bonds have been broken by helicase. Okay, after that happens, another enzyme going to be called RNA polymerase is going to come along, and it's going to start building this piece of messenger RNA. Okay, it's going to match up its complementary strands. So we know that the adenine on DNA will match up with uracil for the RNA. Remember, not thymine, but uracil. Thymine will match up with adenine, guanine with cytosine. So right here we are building our messenger RNA. Okay, just as long as we know that it's going to code with the proper nitrogen base. So we know adenine goes with uracil and so forth, until that particular instruction booklet, that particular messenger RNA gets all of the information on how to make that particular protein. So it won't be the entire DNA strand that this happens, it's just a small segment. When that happens, your messenger RNA is done. Okay, it's been coded, it's taking that transcribed information now and it's built it into this piece of messenger RNA. Once that happens, you have your nucleus, you have DNA, too large to leave. Now you have this small piece of messenger RNA that has the code needed for the protein. Once it does this, that messenger RNA will leave the nucleus. Okay, and it will carry this code in to the ribosome, and how it's put together on the messenger RNA. It's put together in small little sub-packages inside the messenger RNA called codons. And then these codons are going to be read or interpreted by the RNA. So the information from, if I go back, the information here that was built in the messenger RNA was coded in little groups of three package of three letters. So this 3-letter group is a codon, this 3-letter group is a codon, this 3-letter group is a codon, and so forth for the messenger RNA. So UAC is a particular codon, GCU, UGC, and then UAA. Those are all particular codons that are going to be held within the RNA. Now it's ready to leave the nucleus to carry that coded information, that transcribed information to the ribosome. Okay, so let's recap all of this transcription. We know that what transcription is, it's a process where DNA is going to make an RNA molecule that's going to carry that small information out of the nucleus. Enzymes are going to help do this. Okay, helicase is going to unzip the DNA, just like in DNA replication. Then you have RNA polymerase that's going to build an RNA strand that is complementary to one of the DNA strands. Okay, when it's completed, this messenger RNA is going to leave the nucleus. It's going to carry that information in those 3-letter pieces that's going to make up its strand in those codons out of the nucleus into the cytoplasm, where it will then meet up with the ribosome to translate that information.